The right number. Why sample size matters in clinical trials. Imagine a bustling research lab. Scientists meticulously analyze data, their faces illuminated by computer screens. They are on the cusp of a breakthrough, a new drug that could potentially cure a debilitating disease. But before this life-saving treatment reaches the public, it must undergo rigorous testing through clinical trials. At the heart of these trials lies a critical factor often overlooked by the general public: sample size. This refers to the number of participants enrolled in a study, and it plays a pivotal role in determining the reliability and validity of research findings. Choosing the right sample size is like finding the perfect balance. Too small, and the results may lack statistical power, leading to unreliable conclusions. Too large, and the trial becomes unnecessarily expensive and time-consuming, potentially delaying the availability of life-altering treatments. The balancing act: reliability, ethics, and cost in research. Determining the appropriate sample size is crucial for several reasons. First and foremost, it directly impacts the reliability of the study results. A small sample size increases the likelihood of chance findings, making it difficult to determine if the observed effects are truly due to the intervention being tested or simply random variation. Secondly, sample size has ethical implications. Enrolling too many participants than necessary exposes them to potential risks and burdens without any added scientific benefit. Conversely, an underpowered study with too few participants is unethical, as it wastes resources and may fail to provide conclusive answers, potentially delaying medical advancements. Finally, there are practical considerations related to cost and efficiency. Larger trials require more resources in terms of recruitment, data collection, and analysis. Careful sample size calculation ensures that research is conducted cost-effectively without compromising scientific integrity. 5 steps to success. A roadmap to sample size calculation. Calculating the optimal sample size involves several key steps. First, define study objectives. Second, understand data types. Third, select study design. Fourth, consider statistical factors. Finally, incorporate adjustments. Real-world applications, from diabetes to oncology. Let's consider some real-world examples to illustrate the importance of sample size calculation in different clinical trial settings. In a clinical trial evaluating a new drug for type 2 diabetes, the primary outcome might be the reduction in HbA1c levels, a measure of long-term blood sugar control. The researchers would need to determine the minimum number of participants required to detect a clinically meaningful difference in HbA1c levels between the drug and placebo groups. For a vaccine efficacy trial, the primary endpoint could be the prevention of infection. The sample size would need to be large enough to demonstrate a statistically significant difference in infection rates between the vaccinated and unvaccinated groups, providing compelling evidence of the vaccine's effectiveness. In an oncology trial testing a new chemotherapy regimen, the time to tumor progression or overall survival might be the primary outcome. Accurately calculating the sample size is critical for assessing whether the new treatment prolongs survival compared to the standard of care. Tools of the trade software for streamlined calculations. Fortunately, researchers don't have to perform these complex calculations manually. Several statistical software packages, such as SAS and R, offer dedicated modules for sample size determination. These tools simplify the process by providing user-friendly interfaces and a wide range of statistical methods tailored to different study designs and data types. In conclusion, sample size calculation is a fundamental aspect of clinical trial design, ensuring reliable, ethical, and cost-effective research. By carefully considering the factors outlined above and utilizing appropriate statistical tools, researchers can conduct robust clinical trials that advance medical knowledge and improve patient care.